So before we get onto the detail of our research activities in Liverpool, I just want to take a few minutes to take a step back and just talk about how significant health burden are pneumococcal diseases in the UK. So this is a graph that was taken from the annual report released by the Health Protection Agency in 2011, showing that in infants, more than 1/3 of bacterial cases of infections are accounted for by pneumococcal diseases, and about 2/3 in the adult and in the elderly population. 2/3 of the cases of bacterial infections are also accounted for by pneumococcal infectious diseases. Pneumococcal meningitis affects all groups of ages, and with a crushing incidence in the infants and the young children, and in the adults above 45 years old, and in the elderly population. UK is actually probably the second or the third country that has the highest incidence of pneumococcal meningitis in children below the age of 2 and below the age of 5. And it also carries one of the highest fertility rates for pneumococcal meningitis in Europe. Now, what do we know about the causative agent of pneumococcal meningitis? So it is known as Streptococcus pneumonia, or the pneumococcus. It is known to be a gram-positive bacteria with the characteristic lancet-shaped diplococci. And so far, over 90 different serotypes of pneumococci have been reported on the basis of their the structural variations in the polysaccharide capsules that surrounds the bacteria. It is also known to be a commensal, just like the meningococcus of the nasopharynx, and it resides asymptomatically in healthy carriers. However, like Jay mentioned it, under favorable circumstances or in genetically susceptible individuals or immunocompromised individuals, the pneumococcus can turn into an invasive bacterium, spread to peripheral organs, and induce life-threatening diseases such as pneumonia, bacteremia, or meningitis. So what is known about the virulence factors of Streptococcus pneumoniae? Quite a lot of them have been defined. Probably the most well-known and well-described virulence factors is the capsule, so the sugar coat that surrounds the bacteria. Other groups of virulence factors have been described, so as adhesion proteins, like LPX2G anchored proteins, but also colline-binding proteins, which are found on the cell wall of the pneumococcus, and other nutrient receptors, such as metal-binding protein and ABC transporters, which are involved in the trafficking of oligopeptides. But probably one of the most talked about virulence factors in pneumococcus is pneumolysine, which is a toxin that's released is a fascinating protein, in fact, because it's composed of 4 domains, and it has the capacity to undergo conformational changes, as pictured here, and oligomerize to form pores that will then induce lysis of the target cells. So the gold standard up to recently was the use of antibiotics such as penicillin, but with the recent rise in penicillin-resistant pneumococci, the use of alternatives such as vancomycins and cephalosporins is now recommended. So according to a WHO report, the antibiotic resistance featured by pneumococci has now reached such a stage that quite a few of the existing antibiotic classes are now under threat. And we're talking about multi-drug, resistant pneumococci. In terms of preventive strategies against pneumococcal diseases, in the UK, there are currently 2 vaccines that are licensed for use, the Pneumovax 23 and the Prevnar 13, which are both based on polysaccharide subunits that are found on the capsule surrounding the bacterium. In the UK, the vaccination program against pneumococcal disease has evolved quite rapidly in the past 10 years, with the 7-valent conjugate vaccine introduced in 2006, the 10-valent introduced 3 years later, and most recently the 13-valent conjugate vaccine, which was introduced in 2010. Now, while vaccination has undeniably been played a significant role in reducing the incidence of pneumococcal diseases, it presents severe limitations in that it only offers a limited coverage. So, for example, the 13-valent vaccine can only provide protection against 13 of the over 19 potentially pathogenic serotypes. In addition to limited coverage, vaccines also have the introduction of conjugate vaccine has also led to the emergence of a phenomenon known as serotype replacement, which basically refers to an increased incidence of pneumococcal diseases caused by serotypes not included in the vaccine. And this serotype replacement phenomenon is well illustrated in these figures, which again was taken from a report released by the Health Protection Agency, and which shows that upon introduction of the conjugate vaccines in 2006, there was indeed a decline in the incidence of pneumococcal diseases caused by the vaccine included in the 13-valent vaccine. On the other hand, all the serotypes that were not covered in that vaccine have then seen a concomitant and steady increase with the decline in the serotypes contained in the vaccine. So altogether, the goal to tackle antibiotic resistance or to find alternatives to the use of antibiotics and limitations of current vaccines, such as serotype replacement, we're asking the question, and we're actually saying that if you want to develop new and improved vaccine or develop new therapeutic strategies, we need to acquire a better understanding of the host bacterial interaction and try to understand more about what pneumococcal carriage is, what are the mechanisms that maintain carriage, and what are the mechanisms that allow the pneumococci to progress from carriage to invasive. So to address this research question, in Liverpool we've developed in vivo mouse models which aim at mimicking the different stages of a disease. So we've developed a model of nasopharyngeal carriage in mice, where the mice are administered intranasally. N stands for intranasally. Once in the nasopharynx, the pneumococcus will either get cleared or reside asymptomatically in the nasopharynx thanks to a balanced immune response between the host, a balanced immune response within the host. So that's our nasopharyngeal carriage model. We've also developed a pneumonia model, which, based on the doses and the volume that you're giving to the mice, you can allow the progression of pneumococci from the nasopharynx into the lungs. Once in the lungs, again, two outcomes, either clearance or the presence of the pneumococci in the lung will then allow seeding into the blood and cause disease. A third model that was developed was a sepsis model, in which the mice are infected intravenously with the pneumococci. And here again, the bacteria will either get cleared or disseminate into peripheral organs such as the lungs and nasopharynx and cause disease. A more recent model that we've just developed in the past year was the meningitis model, in which mice are administered intranasally with the pneumococci, allowing translocation into the lung and then into the blood, and from the blood into the central nervous system or the meninges. So one of the questions you're trying to answer here is it could be either from the nasopharynx or going into the central nervous system from the nasopharynx or to the blood. And to address what's currently known about the etiology of pneumococcal meningitis, we're also looking at various entry routes into the central nervous system and the meninges. So we're either infecting the mice intranasally, intravenously to mimic what's currently believed to be the entry route into the CNS, so the breaching of the blood-brain barrier, and direct introduction into the, so that's intracisternal, direct injection into the central nervous system, which would mimic a scenario of post-traumatic meningitis, for example. In our cage model, we've provided evidence that pneumococci introduced into mice could be carried asymptomatically in the mice for a duration of up to 28 days, with no detectable level of bacteria in the lungs or in the blood and no clinical signs of disease. Now, what we found that was interesting was that virulence factor that I mentioned earlier, the pneumolizin toxin. When the mice are infected with pneumococci that are deficient for pneumolizin, the stable colonization is no longer there. It becomes a transient colonization with a total clearance at 28 days. This suggests the importance of pneumolizin in maintenance of a long-term carriage. Now, we've done quite a lot of, there has been quite a lot of data gathered on what are the factors that are important in maintenance of pneumococcal carriage, and then some of them have been published very recently. But I'm just going to summarize our current view on what is important in maintenance of pneumococcal carriage. So we believe that upon stable colonization, there is a homeostatic state that exists between a population of immune cells known as T regulatory cells and another population known as effector cells. Now, during a stable colonization, there is a balance here, which is schematically represented here, where the T regulatory cells and the effector inflammatory cells are happily cohabiting. The inflammatory cells will take over the T regulatory cells. The control over the inflammatory response exerted by the T regulatory cells will be lost, and a breach will be created in the epithelial mucosa in the nasopharynx and allow the pneumococci to migrate downwards to the lungs. Once in the lungs, there'll be either two outcomes again. It'll be either a clearance event, thanks to the activity of immunogenous cytokines such as TGF-beta and IL-10, or an uncontrolled inflammatory response, which will lead to further tissue damage and cause seeding into the blood. And in this case, effector inflammatory cells would have taken over and mediate seeding into the blood via cytokines such as interferon gamma. Now, I'm going to get on to pretty much the detail of the research activities that we've been doing the past year and a half, or nearly 2 years now. So when I joined the project, I was presented with genomics data, which initially I didn't understand anything about. I still don't. But anyway. I was presented with data that were obtained from the comparative analysis of 70 pneumococcal isolate obtained from bacteremia patient to 70 pneumococcal isolate obtained from the cerebrospinal fluid, so from meningitis patients. And the analytic pipeline that was developed at the time between the group of Dr. Dean Everett and Sanger Institute, led by Dr. Stephen Bentley, was to, in a way, identify a meningeal virulum, so a set of genes that would be highly characteristics of meningitis-causing isolates. This study then led to the shortlisting of 30 genes. Of those 30 genes, we looked at the function and the expression pattern of the top 5. We've also looked at any risk of sequence homology with human protein, which would rule out their use in human vaccine, basically. Those 5 genes were then subjected to mutagenesis and phenotypic analysis. So those five genes were then used as targets to create mutants. So we used lab strains as well as clinical strains. And we targeted, like one of those 5 genes is PAV-A, for example. It's one of the adherence protein found at the surface of the pneumococci. So what we did was we suppressed the expression of this protein in lab strain as well as clinical strains. And we characterized them for their propensity to be phagocytosed, so engulfed or eaten up by phagocytic cells. So the cells that help you to wipe out any bacterium, any infectious bacterium. And we've shown that when the clinical strain that we've used was deficient in PAV-A, its propensity to be killed by immune cells, so what we want to achieve, was highly enhanced. Similarly, the immune system is composed of a multitude of proteins, and one of them is known as the complement. And we measured the ability of the complement to bind at the surface of those pneumococci, so the wild type, as well as the deletion mutant. And we've shown again that when PAV-A is absent, the ability of the complement to bind to the pneumococci is significantly enhanced. We've also characterized those mutants in vivo. So this is just a picture where PAV-A is located at the surface of the pneumococci. And we've used outbred as well as inbred mice. And the reason for that is that we've had previous experiences with those strains of mice, and we've determined that MF1 mice were, as a comparison to CBA, much more resistant to pneumococcal infection. And CBAs are what we tagged as susceptible mice. So as, let's say, for example, we know now that the immune system is actually involved in immune cells, is actually involved in that susceptibility of these mice. So you could see this as susceptible individuals versus resistant individuals to pneumococcal infection. And we've shown that when the mice were infected with wild type versus the deletion mutant for PAV-A, when PAV-A is deleted, the survival of the mice is significantly enhanced, suggesting the importance of PAV-A as a virulence factor and as a potential target for vaccine development. We've done further analysis in vivo with this deletion mutant for PAV-A, and we've infected mice using our nasopharyngeal carriage model. And we've determined here again that, so in blue you have the mutant, and then in green you have the wild type. And you can see that there is clearance of about half of the mice there at day 10. And statistically significant difference here at day 1 and day 3, suggesting again that PAV-A is important, not just in the virulence of pneumococci, but also in establishment of carriage. And because this was actually developed as a preliminary trial towards our meningitis model, we've looked at the brain homogeneity as well. And again here, the knockout is in blue and then the wild type is in green. And you can see here statistical significance between the wild type and knockout strain, showing that again, when PAV-A is deficient, I made the hypothesis that the olfactory system could be the gateway to the central nervous system, basically. So pneumococci are found in this area in the nasopharynx. What separates the nasopharynx from the brain is basically quite a thick layer of mucosa, which is schematically represented here, composed of a thick epithelium, a cribriform plate, and then an olfactory bulb, which can then go on to the meninges and the brain tissue. And we want to test whether there is a direct translocation from the nasopharynx into the brain. And to allow us to do that, so that's just a description of the meninges, basically. So our hypothesis is that pneumococci can migrate up the neurons, the olfactory neurons, these blue lines here, and into the central nervous system. To test that, we have created some pneumococcus that were bioluminescent. So that's what it is, they're streptomycoccus. They're bioluminescent, meaning that you can image them. So the mice here they're alive, they're just asleep, basically. So that's the beauty of using bioluminescent bacteria, is that you can save on the number of mice, and you can just have the mice asleep, take a picture, put them back in their cage, take a picture a few hours later. And we've shown that. So this is the nasopharynx so that then eventually you'll have to cull the animals and dissect the tissues. And here, this is the tubes where the nasopharynx tissue was collected, and it's very blue because of the bioluminescence of the pneumococcus. But what's interesting here and what is novel is that we have shown the presence of pneumococci in the olfactory organs, olfactory systems. And we've shown these are pictures that were taken at 24 hours, and we showed that even after a week, these pneumococci are still present in the olfactory system. Now, we've correlated these images with viable counts. So all these tissues were homogeneous, and we've counted the number of viable pneumococci. And we've shown that there was a progression into 7 days and a stable, you could almost call it a kind of carriage, I guess, because the mice were healthy in the epithelium as well as the bulb. So this is just adding additional evidence to the hypothesis that the olfactory system is a gateway into the central nervous system. So how do these results fit in the big picture? So here on the top is what we've been working on the past nearly 2 years, and thanks to the support of Meningitis Now. We've used the parent data that were generated in Dr. Dina Red's lab, and we've created mutants based on the list of 5 genes that we thought were candidate genes for vaccine development. We've characterized them. We've looked at their immunomodulatory properties in vitro as well as in vivo. We've tested their virulence. We've created meningitis model mimicking the different scenarios that would lead to meningitis, meningeal infection in human. We're also in the process of doing some proteomics analysis. And we want to take all that further using all the new models and all the new data that we've gathered to validate them as vaccine candidates. So what we're keen on doing and what we're seeking funding for is immunization studies in which all the 5 genes that we've identified here will be tested for their potential to induce antibody responses against pneumococci. We will also optimize the vaccine formulation. We have the expertise to develop adjuvants that will trigger antibody as well as cell-mediated immune response, and most importantly, memory response. And we'll hopefully move forward to our ultimate goal, which is a more effective prevention of meningitis in children and adults. Thank you very much.
Marie, you presented some nice data demonstrating the transit from the nasopharynx to the brain via the olfactory mucosa, which is quite intriguing. Do you know if there's any data to support that for any other meningeal pathogens?
Neisseria. There's evidence that a receptor has been, some glycan receptors has been defined for Neisseria. When it transits from the nasopharynx into the brain, we haven't been able to detect anything in the blood. So we're seeing it. We've actually spoken to, I work with a building full of clinicians. So I basically went to them and asked them, do you see that in human patients as well? And they told me yes. So they have patients which are CSF positive, but blood negative for pneumococci. So we're seeing that as one of the entry route of pneumococci into the brain. We're not saying it's one of the major ones, but it's one of the mechanisms that cannot be ignored when looking at the etiology of pneumococcal meningitis.
Have you shown infection in olfactory neuron cells in culture?
We haven't done it yet, but I'm dying to do that.
The PAV-A mutation
Yes.
and its phenotype, do you have a feel for what actually its role might be that leads to this attenuated phenotype in the infection models?
Well, we know that PAV-A binds to fibrinogen on host cells, so it's it has a role in adhesion. We also know that in a sepsis model, PAV-A mutants cannot seed from the lung into the blood. So PAV-A is definitely important for the transcytosis, I guess, of pneumococci across endothelial cells. And I guess we've shown also with our meningitis model that it is important as well for just the migration or the translocation from the nasopharynx into the brain. So it definitely has to do with altering the adhesion properties of pneumococci. But the other thing that we've shown with the mutants as well is we've looked at the immunomodulatory properties, and somehow when PAV-A is mutated, when the pneumococcal deficient PAV-A, the inflammatory response is enhanced, suggesting that PAV-A might have a more active role than just a binding an adhesion role. It might have more of a role in immune evasion, so helping the pneumococci to remain undetected.
The bacteria in the olfactory bulb, is there any evidence of an immune response?
I'm doing some flow cytometry on that, and there is definitely evidence that there is a recruitment of neutrophil and macrophages into this area when the pneumococci are there. But somehow the mice are still, it's almost like an asymptomatic carriage as well. In the mice, they do not show any signs of disease, so it's almost like it's carried. So it's almost like it's not just a nasopharyngeal carriage, but it's like an olfacto-nasopharyngeal carriage. I guess that's the way I would look at it.
Is there a dose-dependent effect of the number of colony-forming units you inoculate into the nasal passages?
Yes, I do have to use higher numbers. We were trying to mimic what, let's say, environmental stimuli such as particulate materials, like pollutions, or viral co-infections were shown to increase the bacterial density in nasopharynx. And we think that then it becomes a numbers game, that they reach a threshold where the inflammatory response will then create that bridge that's necessary for pneumococci to cross the epithelium and migrate to higher or lower.